But I'll tell you, I didn't have to work. I offered to finish it on Sunday when I started my session 3 days. And I picked it up on Saturday morning before I felt too crummy. By the time Saturday morning, I was like, oh my god. And then Monday, like, the restorations was in. Yeah.
It's already recording this thing. I didn't need to press it. I already did.
But yesterday, I was so sad. I'm telling you, I don't know. Magnesium, I don't know.
Okay. So last time I had been introducing eukaryotic cell, and we didn't quite finish eukaryotic cell. We had gotten to nucleus. I said that the nucleus is this really large organelle, and usually someplace toward the center of a eukaryotic cell, although we know that not all eukaryotic cells have them, because red blood cells don't. But if they're going to be able to reproduce, eukaryotic cells have to have a nucleus. It contains most of the DNA that controls the physiological processes of the cell, determines what proteins the cell can make, so what kind of enzymes, what kind of transporters, what kind of flagella, etc. That nuclear DNA is associated with proteins. Some of them are histone proteins. Those are pretty much homogeneous, which means they're all about the same shape. And then there are other proteins that are enzymes, and they're very heterogeneous, so there's a lot of variety in those. Anyway, the DNA is wrapped around the histone proteins, histone octamers, form nucleosomes, and then the nucleosomes pile up to form this thread-like material that is chromatin. And that's as far as we got. Yes? So we had gone through, and then there was a little movie that I showed you that showed the chromatin forming. Yes? And chromosomes forming. So chromatin can wind up very tightly to form discrete bodies that are the chromosomes. And the chromosomes are what we saw pulling apart with the spindle apparatus when the eukaryotic cell gets ready to undergo any type of cell division, whether it's mitosis or meiosis. Okay, now there is another type of structure that's found inside the nucleus, and it's called the nucleolus. So singular is nucleolus. If there's more than one, they're nucleoli. They aren't always there, and they're not membrane bound themselves, but they're in the nucleoplasm, so they're surrounded by the nuclear envelope. These nucleoli are dark staining bodies, and they show up in some types of cells. You can see them with a light microscope if you have the right kind of cell and if you're really looking closely. So what are these things? They're made up of ribosomal RNA and protein, sometimes called the pacemaker of the cell, but what the nucleolus is actually involved in is the assembly of the ribosomal subunits. So in the eukaryotic cell, our ribosomal subunits are 40S and 60S. Those are made up of our ribosomal RNA and protein, and it's the nucleolus that puts those things together. Ribosomes, which are the site of protein synthesis, the two subunits, the 40S and 60S subunits, those don't come together until a protein is actually being made. And so the nucleolus is putting together the subunits. You have to have the subunits in order to build a ribosome, and you have to have ribosomes in order to build proteins. That's why sometimes the nucleolus is said to be the pacemaker. Okay, so it's the site of ribosomal RNA synthesis, which just means it's a particular region of DNA that is coding for ribosomal RNA, but then it's also assembling the subunits, putting the ribosomal RNAs together with proteins. This is kind of a diagrammatic representation on the left, where you see the nucleus with the nuclear envelope, and the nucleolus is just this dark staining body. And then the photomicrograph, which is on the right, you can actually see the nucleolus. Again, it's a dark staining body. It doesn't have a membrane of its own. But again, oftentimes we think of organelles as being these structures within eukaryotic cells that have specific functions. I would argue they're not necessarily all membranous. And we've seen some that are, and some that are not. Now, another structure that can be found inside the nucleus of a eukaryotic cell is called a spliceosome. Again, soma means body, so then we have to think about, well, what does the splicing have to do with? Spliceosomes are involved in a process called post-transcriptional modification. So when RNA molecules are made, there are regions that are removed. The regions are called introns. Introns get removed, and regions called exons get hooked together. So the cutting out of the introns and the splicing together of the exons, that involves spliceosomes. They are protein and RNA. The RNA in this case is short RNA or sRNA. I'm not sure how many of these things exist, and they're definitely not something that you can see with a light microscope. So these are tiny little structures. We'll talk about spliceosomes more when we talk about how the DNA is processed, so DNA expression. Talk about DNA is used as a pattern to make RNA, and then the RNA has to be modified. It's eukaryotic cell. I just realized you guys don't have your stools. I forgot to go get them.
I'm okay.
Okay, I'm sorry. It's like, oh, we gotta go get our stools.
So there are exons and
So exon stands for expressed regions. The exons are going to be kept. So what the spliceosomes do is cut out the introns. Intron stands for intervening regions. The introns are removed and the exons are spliced together. I mentioned the term ribozyme when talking about ribosomal RNA, that the ribosomal RNA is actually responsible for catalyzing the formation of peptide bonds during protein synthesis. So it is a ribozyme. Apparently the small RNA is also considered a ribozyme because it's catalyzing the formation of phosphodiester bonds between the exons, hooking them together. So this is showing a spliceosome and an RNA molecule. That is not a picture that is taken with any kind of microscope we have access to. So we're really looking at tiny little portions of material there, an RNA molecule and a spliceosome, to give you an idea of the scale on that thing. And then eukaryotic cells can contain inclusions. Usually these are referred to as bodies that are non-living. There's some kind of material that has accumulated inside the eukaryotic cell, and there are a variety of different types of materials that might be accumulated. Sometimes cells form inclusions if they've been infected by viruses. So we're not going to get into that particular category of inclusion, but you can see little pockets that might be oil accumulations or polysaccharide accumulations or maybe even protein accumulations. They just show up in the cytoplasm. Usually inclusions are not designated as having any specific function. They're kind of just there. On the other hand, I often think, yeah, they probably do have a function and we don't know what it is. The cell might be just storing some kind of material, but then they might be able to use it later. And then eukaryotic cells, some eukaryotic cells, are equipped with another layer of material. This is outside the cell membrane, and it's either called a cell wall or a skeleton. So these are rigid layers. Again, they're outside the cell membrane. They're only present on certain types of cells. And for some reason, people that studied plant cells decided that if there was a layer outside their type of cell, that was called a cell wall. And people that studied animal cells, if there was a layer outside the membrane of their type of cells, they called those skeletons. So are they really different? They're spelled differently. Wall versus skeleton. So what types of cells have these? In the plant-like category, algae have walls. I say plant-like because algae have green chlorophyll pigments like plants. Fungi have walls. Fungi used to be considered plants. We now know they are not, definitely not plants. Protozoa typically have skeletons because protozoa are considered to be more animal-like. So we have these different layers. They have a function, a couple of different functions. One, they give a cell a characteristic shape. We often think of this characteristic shape as being significant because we use them to distinguish one cell from another. So you can look at a cell and say, oh, that cell is this shape, therefore it's a so-and-so. For the cell itself, that shape may influence whether it catches water current or air currents that can move through the environment. It may influence whether that cell is subject to predation, so they give it a shape. They can provide protection. Protection might be against changes in osmotic pressure. We talked about eukaryotic cells. If you put them into a hypotonic environment, they either have to have a way to get rid of the excess water, or they have to have something to prevent them from blowing up. Cell walls do that quite nicely. So you can put algae cells or fungi cells in fresh water, they don't blow up. The membrane goes up against that wall, and that's as far as it's going to expand. The skeletons are more likely to be protection against some type of predator. So these things have different compositions. Cell walls are typically made of polysaccharide. Very often when we think of a polysaccharide cell wall, we think of cellulose. And this is because cellulose is the most common cell wall material in plants. And it's what wood is made out of, it's what paper is made out of. So it is a polysaccharide, cellulose. But there's other types. There's chitin, there's glucan, there's pectin, and there's agar. And we recognize the name agar because it's the solidifying agent we use in our microbiological media. But agar is a polysaccharide, and it's found in the cell walls of certain types of algae. Those are just some examples. Sometimes cell walls are extremely thick. The other thing that might be present in a cell wall is glass. So these are the cell walls of organisms called diatoms, and diatoms have quite a lot of glass in their cell walls. This is a type of algae that has more of a polysaccharide type cell wall. Here the cell walls are relatively thin. Here the cell walls are extremely thick. This type of cell has a very, very heavy cell wall. And here, these are fungi, they have cell walls, but it's really hard to see them. So this layer varies considerably in thickness. And it is a cell of characteristic shape, and it does provide protection. Skeletons, again, usually associated with animal-like single-celled organisms, protozoa. So skeletons can be made of calcium carbonate. They also can be made of glass. We're going to be looking at glass skeletons when we look at radiolarians in here. These are some skeletons. Those are calcium carbonate. Those, I think, are also calcium carbonate. And these are glass. So the last one's up. That's a group of protozoa called radiolarians. What you're looking at are just the empty skeletons. So sometimes these things kind of look like tiny little seashells. Sometimes they look like these glass, almost like architecture. They look like a dome on a roof or something like that. And the organism lives inside of that skeleton, and it's protected by that skeleton. And these things can grow as the protozoa gets bigger. They can add to their skeleton. This is a summary of eukaryotic cell. It'd be nice if we had some sound. Here we're looking at a human white blood cell. And it's binding to the surface of the blood vessel. This is what white blood cells do. And then what they're doing with this movie is showing you what kinds of things are going on inside that white blood cell. So we've gone down through the cell membrane as we're looking back toward the outside. And inside the cell membrane, there is this matrix of actin. So these are actin the surface. They're going to gather on a lipid raft that is floating on the surface of the membrane, and then they're going to extend upward. This is showing the contact, again, between the white blood cell and the inner lining of the blood vessel. What happens to the white blood cell then? It actually changes shape, and it goes through between the blood vessel cells, and it goes into the intracellular fluid. And this is why we have fewer white blood cells in the circulation. They don't stay there. We actually produce more white blood cells than red, but our blood is red because the white cells weep, and they're constantly going out into the intracellular fluid. That's one of those kinds of movies. You can find that on YouTube. It's fun movies. And there is a version of this that has narration beyond what I'm telling you.
This mammalian cell has been in.
No, stop. That was the mitochondrion thing. Okay, so that's our eukaryotic cell. Again, we went through that fairly quickly because most people have seen eukaryotic cell information before if you've been through anatomy and physiology, because human cells are eukaryotic. The next cell category we're going to be looking at are prokaryotic cells. And the prokaryotic cells, a lot of people have not seen. I have to figure out where I left them. The good news when we're looking at prokaryotic cells is they're not very big, and they're also not very complicated. However, I just had a realization. I left my prokaryotic cell on the other side of the building. I'll be right back.
This is the key from the previous and last year's class. Controls. If you look at the guides.
Okay, so this is our pet prokaryotic cell. It's a nice big one so that you can see it nicely. And if you looked at this prokaryotic cell and you were thinking eukaryotic cell, you might see some structures that are real similar, right? Like these things. What do you suppose those are? Those are flagella. And you might think these are cilia, but they're not. This is something you need to know. Prokaryotic cells do not have cilia. So we're going to let this be our model of a prokaryotic cell. They do have some features in common with eukaryotic cells, but again, they're much more simplistic. As I said, if you think about the eukaryotic cell as being a mansion with all these different rooms, and in each room there's some specialized function going on, the prokaryotic cell is like a tent. You're not going to have membranous organelles in here. This is not going to be compartmentalized. It's a very simple type of an organism. But they do have flagella. So here we see these structures extending out, these long ones. These structures that here might look like cilia are not. Again, prokaryotic cells don't have cilia. These things are called fimbria. Actually, fimbriae when there's numerous. What we're going to be doing with this prokaryotic cell is looking at it starting from the outside and kind of working our way inward. We know that cells, all types of cells, have cell membrane, because we talked about the membrane and membrane function and so forth. So in this case, we're going to start on the outside, work our way into the cell membrane, and then go from the cell membrane into the interior. Okay, so the first few structures we're going to be looking at are outside. Let's put that guy down. So, outside as in external to. What do we find outside the cell membrane or external to the cell membrane on a prokaryotic cell? Okay, so the first structure, which was visible here on this model, these long, slender, whip-like structures, are flagella. Eukaryotic cells had flagella also, but we're going to find out prokaryotic flagella are very different. Again, flagella is plural. If you have only one, you have a flagellum. Since there are three of these are flagella. But these things are made of proteins called flagellin proteins. And the flagellin proteins are little units that then get connected together to make this very complex structure. So here you can see flagellin proteins. Here you're looking at a kind of a cross-section of these flagellin proteins, and you can see that there is a hollow core. The interior of the flagellum itself is hollow. This thing is very different than the structure of the eukaryotic flagellum. So when we were talking about eukaryotic flagella, they had a skeleton made up of what? Microtubules. Microtubules in 9 groups of 2 with 2 in the middle. And eukaryotic flagella are surrounded by the cell membrane. This is not true of prokaryotic flagella. Prokaryotic flagella are made of these proteins, flagellin proteins, so they're not surrounded by the cell membrane, they're not supported by microtubules. Again, here we see the flagellin proteins. You can see this flagellum on the right-hand side connected to the surface of the cell. Well, this flagella on the prokaryotic cell are connected to the cell membrane. They're also connected to the cell wall. So I've just got finished talking about eukaryotic cell walls. Prokaryotic cells typically also have a wall. It's going to be different. It's still a layer outside the cell membrane. So typically the flagella are connected to both of these layers, but not surrounded by the cell membrane. Each flagellum has a set of rings. The number of rings is variable depending on what type of cell you're looking at. Tonight we're going to do gram stains, and you're going to learn about the difference between gram-positive bacteria and gram-negative bacteria. Their walls are different, and consequently the structures that connect the flagellum to the wall are going to be different. But it's kind of interesting because the flagellum is kind of built like an axle with a series of wheel-like structures. We'll call them rings. The axle is connected to the wall and the membrane by these rings. And these flagella spin. So here we see this structure. In this case, we're looking at, this is a gram-negative type bacterium. And the reason I can say that is because this bacterial cell, here we're looking at a cross-section. We have an inner membrane and we have an outer membrane. Only gram-negative bacteria have 2 layers of membrane making up their cell surface. So this is the cell membrane. This part we're calling the inner membrane down here. This is the cell membrane. The cell wall material, the rigid wall material, is peptidoglycan, and then there is an outer membrane. So here we see that we've got a fairly complex ring-like structure associated with the cell membrane, and then we have rings associated with the wall and the outer membrane. This portion that's down in here is the motor. So another function of prokaryotic cell membrane is to provide the energy, if you will, that drives the movement of the flagellum, and the flagella spin. So they have rotary motion. The other thing about these flagella is they tend to be very long relative to the length of the cell. They're extremely fine. You cannot see bacterial flagella with a light microscope unless you stain them. And basically what that means is you use some type of a material that sticks to them and makes them fatter, because otherwise you can't see them. We can see eukaryotic flagella without staining, but not prokaryotic flagella. Here we're going to see, again, this set of rings associated with the cell surface. Here's our gram-negative cell again. This is the cell membrane, and then there is an outer membrane, and this material in between is the cell wall made of peptidoglycan. So we have rings here, and here, and here. In the gram-positive cell, we have a set of rings associated with the membrane. Those are, again, the motor that's going to drive the movement of the flagellum. And what this arrow here indicates is that this flagellum is going to spin. The flagellum typically kind of comes out of the cell, makes a 90-degree bend, so that when that thing spins like an axle, it causes the flagellum to move. Eukaryotic flagella movement was whip-like, down, back, down, back. Prokaryotic flagellum is like a wheel on an axle. It is rotary motion. I think humans invented the wheel. Not so much. Bacteria were using wheel-like structures for a long time. So they spin. And again, usually the power that makes this thing work is an ion flow. It can either be hydrogen proton flow or sodium flow. There you see a diagrammatic representation, again showing you that motor, which is a set of rings associated with the cell membrane. And then we're going to watch an animation. What's another thing that's kind of interesting about a prokaryotic flagellum is the spin. The direction of the spin is reversible. So they can spin in one direction, or they can reverse and spin in the other direction. The result of that is the cell can tend to go in one direction, or it can go back in the other direction. So they're not consistent. Here we see the flagellum moving. And if we look closely, we see this thing is spinning. And again, the spinning is being driven by this motor of sorts that is associated with the cellular membrane. The little particles that you see moving there, those are either hydrogen or sodium ions, and that's what's providing the energy to make that thing spin. What's interesting is sometimes bacteria have these things all over their cell surface. Somehow they have them all spinning and they don't get tangled up. It's like, how do they do that? I don't know. Pretty amazing. Now this is not an animation. This is actually a film of bacteria spinning or swimming. So they're using flagella. Again, if you see bacteria swimming, they have to use flagella. They do not have cilia as structures to make them swim. Watch for a characteristic movement of bacteria. They'll do a run, which means they're going in one direction. And then if they stop and kind of spin around, that's called a tumble. And they will often orient themselves to go toward a particular food source. They want to show positive chemotaxis. And let's say the food source is the clock over there. They might be going this way. They'll stop, tumble, 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 and go this way, tumble, tumble, and finally get oriented in the right direction. And then they just go. They stay in that orientation. So we're going to watch them swim pretty quick. This is actual speed that you can see something like this. We will be doing wet mounts in here so you can watch bacteria swimming. Notice they're not all going in the same direction. Bacteria don't swim in formation. They're going all different directions there. Some of them aren't swimming at all, and this is kind of characteristic if you make a prepared slide of bacteria. Some will swim and some are just, they may be stuck to the glass. I don't know why they're not swimming. Some are lazy. Some might be dead, I don't know. Okay. Now, if we were in a situation where we could do flagellar stains, and again, a flagellar stain means you're going to stick some kind of material on the outside of the flagellum to make them fatter, then you could do some identification of bacteria based on how many flagella they have. A lot of times, reference volumes will tell you the bacteria have one flagellum or they have multiple flagella. Well, so there's terms that have to do with this flagellar number and arrangement. And the root of the word is trick. Trick means hair-like, so the flagellum is kind of a hair-like extension coming off the cell. If the cell is atrichous, remember A means without, so if they're atrichous, they don't have any flagellum. If they're atrichous, they can't swim because bacteria don't have any other means of swimming. They don't have cilia. So if they're atrichous, they're not going to be swimming at all. Unfortunately, a lot of our morphological unknowns are atrichous, and students get kind of bummed out. It's like, oh, mine can't even swim. Yeah, I'm sorry. Some of them can't. If they are monotrichous, mono means one, as in monosaccharide, one sugar unit. So mono means one. If they're monotrichous, they only have one flagellum. I don't know how many bacteria are monotrichous. If they are paratrichous, para means around. But if they're paratrichous, they're also polytrichous, which means they have many flagella. Para meaning around, it means the flagella are fairly evenly distributed over the cell surface. So they'll have flagella all around. And if they are amphitrichous, amphi we learned in association with amphipathic, amphiphilic. Amphi means two ways or both ways. And what it refers to when you're dealing with flagella is that you have flagella at both ends. You may have one flagellum at each end, or you may have flagella in tufts at both ends. Sometimes those are called lophotrichous. But we have these slides, amphitrichous and paratrichous. So you will have an opportunity to look at flagellar prepared slides in the laboratory, and you need to be able to recognize paratrichous versus amphitrichous. So para, again, means flagella all around. This is a cell. You see these little hair-like structures just coming off in all directions. That is, wow, that's Paratricus flagella arrangement. Sorry, I'm just noticing that the autoclave is exhausting, and so this corner of the room is starting to smell really bad. Like, whoa. If they're Amphitricus, they have flagella coming off both ends. Here you can see the flagellum on this end. Wow. Well, there should be flagella coming off that end too. It's kind of hard to tell. Anyway, Amphitricus flagella at both ends, Paratricus flagella all over the place, all over the surface. Okay. Okay, we're still outside the cell membrane, so we've got flagella. That's our first structural type. The next structure we're going to be looking at are these fine hair-like structures. So the flagella are kind of hair-like, but they're heavier than these things. These are really fine. And again, they are not cilia. So we're going to be looking at structures called fimbriae and pili. For a long time, microbiology textbooks were not real clear about whether these were distinctly different structures. So they kind of use the terms interchangeably. As more time has gone by, more accurate identifications have been made, and apparently these are definitely different. They're made of different types of proteins, so they're not the same kind of structure. The fimbriae, so if you see the IAE ending, that means it's plural. If it's just one, it's a fimbria. But anyway, these are fimbriae because they're very numerous. So there are many of these on the surface. We know they're fimbriae because fimbriae tend to be very numerous, whereas pili, usually there's only one, or maybe two, but they're not all over the thing. So these are fimbriae, and they're made of fimbrin proteins. If they were pili, singular pilus, these tend to be longer, although as soon as I say that, I think that's not necessarily so, because pili can extend way out, and they can also shrink. So their length is quite variable, but there's usually only one, sometimes two. So what are these for? They are not for locomotion. That is, they're not used for swimming. They don't have the capability of using a whip-like motion with either fimbriae or pili. Instead, they're used for attachment. In some cases, the fimbriae can aid pathogenicity as well, and we'll see why. These are electron photomicrographs. Some of them have been artificially colored, but where you see a lot of these little hair-like structures, they're fimbriae. So all of these are showing fimbriae. This one up here, you see a flagellum also. See how long that is relative to the cell surface? And then these fimbriae are much shorter. These are all fimbriae on the lower structures. So they're used for attachment, and they can attach cells to a variety of surfaces. Let me close this door. Once again, Sasha locks that door, so if I don't take the time to unlock it, it's locked. If you go out, if somebody needs to go out, I'll go out there and unlock the door. Anyway, so the fimbriae are for attachment. Cells can attach to wood or rock or leaf surfaces, or they might attach to a host cell with their fimbriae. Pili are typically used to attach cells to another cell of the same type. And they use these things to pull the two cells close together so that they can exchange DNA. If they have that function, they're typically referred to as F pili, F standing for fertility, or sex pili. And old textbooks used to say the pili were hollow and that DNA was transferred from cell to cell through the pilus. Apparently that's not the case. Instead, the pilus extends out, touches one cell. So here we see an example. If this is the cell producing the pilus extends out, makes contact with the other cell, and then shortens, and the cells get pulled close together. Once they have gotten close enough, then DNA exchange can take place. But the DNA doesn't actually go through the pilus. It is, again, this is the function of sex pili. What's kind of interesting about this is I don't know of any other pili. So there may be some other pilus type, The only ones I'm familiar with are sex pili. So they're involved in gene transfer. Okay, moving to our next type of structure that's found outside the cell membrane. In this case, since our model doesn't have one, so we've got flagella, we've got fimbriae, we don't have a pilus. If you could imagine covering this cell with something, maybe the texture of whipped cream, I couldn't put it on there and make it stay. It would gradually melt and fall off. But whipped cream or silly putty or something kind of sticky-like, then we would have our next structure, which is called the glycocalyx. Think about glyco. Glyco, when you're talking about glycogen, glycogen is a polysaccharide. So glyco refers to polysaccharide, and very often the glycocalyx is made of polysaccharide. It may sometimes contain protein, but it's a layer outside the cell wall. So again, we're starting from the outside, moving toward the membrane. The glycocalyx is outside the cell wall, and it tends to be made when cells have a period of time where there's a lot of nutrient available to them. A lot of times bacteria, their life is more feast and famine. So if they're feasting, they can put a lot of material into storage on the surface of the cell. Then if there's no food available to them for a long period of time, they can use that stored food. So one function of the glycocalyx is a food reserve. Here you can see a glycocalyx this is what's called a capsule stain, where the cells are these little purple guys, and this material around the outside is the glycocalyx. This glycocalyx is pretty thick. Sometimes they're even thicker than that. On the other hand, sometimes they're much thinner, and they kind of come and go. So the cells, just because a cell can produce them, doesn't mean you're going to be able to see them. They're going to be produced when there's a lot of nutrient available. If the glycocalyx is dense and well organized, we do call it a capsule. So what you saw in the image, that was a capsule. It was a distinctly definite layer outside the cell surface. It's well organized, it's a capsule. If it's loose and poorly organized, it's called a slime layer. And you can imagine slime is not so well organized. It's going to be a loose layer. A lot of times you'll see slime, depending if you do a certain type of stain preparation, you may be able to see the slime as a kind of linear material. It looks like it's kind of washing off. If bacteria are making a glycocalyx, oftentimes their colonies will look glistening. So remember Klebsiella, when we were describing the surface texture of Klebsiella colonies, and I said they really are glistening, wet looking. That's because Klebsiella form capsules. And so here you can see an image of bacteria that are capsule forming. The colonies themselves look wet and slimy. Some bacteria, remember how we incubate our agar plates, agar side up? So if the bacteria produce a slime layer, often what they'll do is they'll start to reproduce and they're hanging on the agar. They will get so wet with that slime that the slime will drip into the lid. They can get really messy. So, yeah, lots of fun with bacteria. So, functions of the glycocalyx. Primary function is nutrient reserve. It's food storage. Again, they build a glycocalyx when there's plenty of nutrient, and then they can use that later on when there's less food available to them. But that's not the only function, because the glycocalyx is sticky, and it's going to allow the cells to attach to a surface that might be really slick. What we're seeing here in the image, those are teeth, and bacteria forming a glycocalyx. This happens on the surface of your teeth all the time. So if you brush your teeth regularly, and then you miss a day for some reason, maybe you're off someplace and your toothbrushes are with you, and then maybe you take some dental floss and you move it over the surface of your teeth and you get this white gooey stuff on there, that's glycocalyx. So we have bacteria that live within our mouth. Streptococcus mutans is very common bacteria, and they form this layer of material on the surface of the teeth. And you can actually see it on your dental floss. Hard to see it on a toothbrush, and so here you see it building up on the lower teeth. If it gets calcium in it, it forms plaque, and that can do quite a lot of damage. Another function of capsules is to provide protection. So oftentimes capsules are associated with the formation of biofilms, the cells that are living in a biofilm are protected. They're protected against desiccation, they're protected against toxic chemicals, they're protected against predators, and this is quite problematic if the cells are potentially pathogenic and they are taking up residence on the inside of some type of tubing used in a clinical setting. There's quite a lot of tubing used in clinical settings where that tubing is running liquid through, and if the bacteria get in there and they form biofilms, it's really hard to get them out of there. So again, this is a protective aspect of the biofilm. The other kind of protective feature of capsules, for some reason, the polysaccharide material that makes up the capsule is not something that our white blood cells recognize as something they should eat. So usually the role of our phagocytic white blood cells is to find bacteria and consume them, get rid of them. So they eat them, phagocytosis. If the bacteria have a capsule, oftentimes the phagocytes just ignore them. They won't eat them, and consequently, bacteria that produce a capsule are much more likely to be disease-causing agents because your white blood cells don't destroy them. They get to stay there, they get to reproduce, and that can be very problematic. Turns out that there are different strains of Streptococcus pneumoniae, and it's the Streptococcus pneumoniae that produce capsules that are highly virulent. They're highly likely to cause disease. If they don't have capsules, your white blood cells just eat them, and they don't get to stay around and reproduce. So they can aid pathogenicity. Here we see Streptococcus pneumoniae. This is a capsule-forming strep, and you can see the capsule is quite large, so it's giving that cell quite a lot of protection. Okay, so we've got our glycocalyx. We're still moving inward toward the cell membrane. The next structure that we're looking at is the bacterial cell wall. We're going to spend quite a bit of time in lab talking about cell wall composition because we are doing Gram stain today, and the Gram stain is a stain based on cell wall composition. Again, this is a rigid layer, which is something we have in common with eukaryotic cells. Cell walls of eukaryotic cells are also rigid, and they're found outside the cell membrane. So eukaryotic cell walls are also outside the cell membrane. Prokaryotic cell walls are unique, specifically bacteria. The cell walls of bacteria contain a unique material called peptidoglycan. Again, glycan tells you there's carbohydrate associated with it, but what does peptide? Where do we see the term peptide?
Protein.
It's the bond between the amino acids that make up the protein. So if you have a string of amino acids connected together, you have a polypeptide. So peptide refers to a string of amino acids. That tells you that the cell walls of bacteria are made of strings of amino acids and polysaccharide. Peptidoglycan is unique to bacteria. No other types of organisms have that material. So we spend a bit of time talking about it. The peptidoglycan is a rigid layer, and on bacteria that we call gram-positive, the peptidoglycan layer is very thick. It can account for more than half of the cell's dry weight. So it is a very thick layer. Again, it's providing protection to the gram-positive type cell. On a gram-negative cell, the peptidoglycan layer is thin, and the gram-negative cells have another layer of membrane outside their peptidoglycan layer. So the wall composition is rather different, and this influences some physiological aspects of gram-positive and gram-negative cells. In these images, the cell membrane is here. This is a gram-positive type wall. So here's the cell membrane, and then we have multiple layers of peptidoglycan, so that it's quite a thick wall. There's some other things in there, some teichoic acid molecules, which we'll get to later on. We'll talk about cell wall composition in the lab. The gram-negative bacterial cell, here's the cell membrane. Here's the peptidoglycan. It's very thin. And then outside of that is another layer of membrane. The composition is different from the cell membrane, but it's still another membrane layer. So how does this influence their physiology? The gram-positive bacteria with their thick peptidoglycan walls tend to be much more resistant to physical damage. Sometimes people get confused about physical factors versus chemical factors. Physical factors are things like temperature differences, pressure, radiation. So the gram-positive bacteria with their thick walls are more resistant to damage caused by exposure to heat. They can tolerate higher temperatures. They're resistant to radiation. They're resistant to desiccation, much more so than the gram-negatives. And pressure, whether it's osmotic pressure or some other type of pressure. The gram-negatives, because they have that outer layer of membrane, that outer layer of membrane tends to prevent chemicals from coming through, and so they tend to be more resistant to chemicals. There are certain chemicals that are used to control bacteria, such as basic dyes, beta-lactam antibiotics. The gram-negative bacteria are more resistant to these. The gram-positives tend to be more susceptible. Also, an enzyme called lysozyme. Lysozyme breaks down peptidoglycan. So if you put lysozyme on a gram-positive cell, it takes up the peptidoglycan apart and they don't have a wall. But if you put it on a gram-negative cell, probably it can't get through that outer membrane, so it's not going to do the same kind of damage. Now, there's another structure that's found outside the cell membrane, but inside the wall, or at least inside part of the wall. Depends on what kind of cell you're looking at. This is called the periplasmic space. Again, peri, P-E-R-I, means around. So we're talking about a space that is around, it's around the cytoplasm. It's outside the cell membrane, but it's inside at least a portion of the wall. Depends on what type of cell you're looking at. And it's really not an open space, it's a potential space. So if we look at a diagrammatic representation, here again, this is the gram-negative cell wall, where you have cell membrane, and you have outer membrane of the wall, and here's your peptidoglycan. The periplasmic space is basically the space between the inner membrane and the outer membrane. So on a gram-negative cell, the peptidoglycan is kind of in the periplasmic space. It's just a space. Now we said that the cell membrane of prokaryotic cells is involved in synthesis of wall components. As the wall components are synthesized, they're moving into this space. So you can think of the periplasmic space as being for storage. There can also be enzymes accumulating in there. But it's not a real big gap. It's not even as big a space as you see on this diagram. Now, certain types of bacteria have flagella within their periplasmic space. So these are called periplasmic flagella. Unfortunately, they're also called endoflagella or axial filaments. It's like, really? Come on, biologists, can you just come up with one name and stick to it? We're going to call them periplasmic flagella because they're in the periplasmic space. And as far as I know, the only bacteria that have these structures are spirochetes. Spirochetes are very slender, long, spiral-shaped bacteria. Here you see images of spirochetes. They're pretty hard to see with a light microscope, and these are electron photomicrographs. The one on the left is an illustration. So what it's showing you, this would be the cell, and these flagella are connected to one end of the cell, and then they're extending back and wrapping around the cell, and they're inside the outer membrane. Spirochetes apparently have an outer membrane, so they have the cell membrane, and they have their peptidoglycan layer, and then they have their outer membrane. And these periplasmic flagella are in that space between the peptidoglycan and the outer membrane. What's tricky is, again, think about prokaryotic flagella, they spin. And they spin, and they're in that space. When spirochetes move, they move like a corkscrew. The whole cell spins. And apparently it is because of the way the flagella spin inside there. So only spirochetes have periplasmic flagella. And they're just flagella inside the periplasmic space. So again, we've been moving from the outside toward the cell membrane. We finally get to the cell membrane, and now we can review some of the functions. So it's the layer that surrounds and limits the cell. It separates the protoplasm from the external environment. It regulates what gets in and what gets out when you think about transport processes. simple diffusion, facilitated diffusion, osmosis, active transport, primary, secondary, etc., etc. When you get to prokaryotic cells, the cell membrane is supposedly not engaged in endocytosis, but there have been some experiments to show that if you give them the right proteins, they can. And I'm going to give you an example where I think they're actually doing endocytosis. And then the cell membrane of the prokaryotic cells has ATP synthase, an important enzyme involved in making ATP. And sometimes the molecules that are associated with trapping light energy, so bacteriorhodopsin is a light trapping pigment, and there are chlorophylls in cyanobacteria. And of course, the receptors necessary for quorum sensing, and the receptors necessary for taxis, and all those different functions associated with membranes. So they all have to have it. Sometimes you'll see structures that are described. So cyanobacteria, this is a particular type of prokaryotic organism. The cyanobacteria I mentioned last time because I said chloroplasts evolved from cyanobacteria. So these are a specific type of bacteria. They're capable of capturing light energy using chlorophyll molecules, and the chlorophylls are associated with these inner membranes called thylakoids. So the specialized membranes that are associated with photophosphorylation, they have the ATP synthase. As far as I know, thylakoids are only found in cyanobacteria, even though some of the prokaryotic cells do photophosphorylation. The other thing about thylakoids, I believe they're just folded regions of the membrane, the cell membrane. Some references suggest that they're actually separate little organelles, if you will. We typically say prokaryotic cells don't have organelles though. So this is showing that folded membrane. Again, this is the site of photophosphorylation. So they have quite a lot of folding, so they have a lot of surface area for making ATP and trapping light energy. There are structures called mesosomes that are mentioned in some microbiology texts. Most recently, it seems that most investigators concur mesosomes don't actually exist. So you think, well, what, do they just make these up? Apparently not. Apparently these things showed up as some material was being prepared for electron photomicrographs. So they would see them, these folded regions, and you can actually find diagrams of these, and sometimes electron photomicrographs that show them. Our cell here has these inner folded regions coming in. These are supposed to represent mesosomes. They're artifacts. They don't naturally occur. So never mind. Apparently there's no such thing as mesosomes. Even though you can find pictures of them, images of them, they're not real. They're just something that shows up because of the way the cells are prepared. So thylakoids are real, mesosomes not so much. So now we're going to go inside the cell membrane. We've gotten to the cell membrane. We know a lot about that. Now we're going to go into our prokaryotic cell. Well, okay, so I can open the prokaryotic cell again. Notice the top part of this. That's like, aside from the mesosomes, which don't really exist, there's nothing in there. The bottom part of the cell, you see a couple of structures. There's some little dots over here. There's this thing. And there's a lot of open space. Again, the prokaryotic cell does not contain a bunch of membranous organelles. There's no endoplasmic reticulum. There's no Golgi apparatus. There's no mitochondria or chloroplast because those are prokaryotic cells. There's no lysosomes, no peroxisomes. So it's pretty simple. That's good in the sense that if you're trying to learn prokaryotic cell structure, you can say, good, we don't have to learn all those things. There are some different things though, wouldn't you know? So what's in there? Cytoplasm. The eukaryotic cell, once you came inside the cell membrane, you had cytoplasm, and then you had nucleoplasm surrounded by a nuclear envelope. The prokaryotic cell doesn't have a nucleus surrounded by a nuclear envelope, so you just have cytoplasm. So all the protoplasm inside the cell membrane is cytoplasm. You don't have cytoplasm and nucleoplasm. You don't have this internal structure. there is that thing that kind of looks like a shoelace in the illustration and looks more like a shoelace in the model because I think it is. So we have that thing in there, but it doesn't have a membrane around it. Oh, the question here is that cell gram-positive or gram-negative? Well, so if you look at this, if you look at the labels on this diagram, what you would see is that there is an inner membrane, it's green, then there's a cell wall in orange, and then there's an outer membrane that is this kind of brighter orange or brownish. If you've got a cell membrane and an outer membrane, it's a gram-negative cell. Only the gram-negatives have 2 layers of membrane surrounding them. Okay, so that shoelace-like structure in this model is showing up only on this portion of the cell. This thing is called the nucleoid. Remember how I told you guys, watch out for spelling, because we've got nucleus, and then tonight we've got nucleolus. We had nucleus and nucleosomes last time. Now we have nucleolus and nucleoid. Watch the spelling. The nucleoid is also sometimes called the nuclear region. The word literally means nucleus-like. Why is it not a nucleus? Because it doesn't have a membrane around it. So it's not separated from the rest of the cytoplasm by a nuclear envelope or membrane. It does contain DNA. This tells you that it's an extremely dense accumulation of covalently closed circular DNA. And that's true for many, many prokaryotic cells, but not all of them. Apparently, bacteria in the genus Streptomyces have linear DNA. Most of the references to prokaryotic DNA will tell you it's covalently closed circular. Usually it is, but sometimes it isn't. There are small amounts of RNA and protein associated with the DNA, which makes sense. Most of the RNA associated with that nucleoid is ribosomal, and cells are constantly making ribosomal RNA so that they can make ribosomes. Some of the proteins that are there are enzymes. Apparently bacteria don't have histone proteins. Archaea, which are also prokaryotic, do have histone proteins. And they also don't have a true nucleus, they just have a nucleoid. So no nuclear envelope, that makes it not a true nucleus. It does contain most of the cell's DNA, and so it's like the nucleus of a eukaryotic cell. It's going to be controlling the physiological processes of the cell. DNA determines what proteins a cell can make, and therefore what kinds of enzymes, what kinds of proteins involved in transport, what kinds of flagellins, what kinds of pillins, all those kinds of things. Again, bacteria don't contain histones. They do contain some other proteins that are pretty much homogeneous, and archaea do have histones, at least some of them. And again, most of the RNA in the nucleoid is, well, I said ribosomal. Most of it is messenger RNA. I believe most of the RNA in the cell is ribosomal. But messenger RNA is also being made in association with the DNA, and it's going to tell the cell what kind of protein to make. And that's the kind of detailed question I'm not going to ask you. I will ask you what is a nucleoid and what is its function. Never mind which type of RNA is most common in there. Then we have small extra chromosomal loops of DNA called plasmids. So as I said, most of the DNA of the cell is in the nucleoid, but not necessarily all of it. So a plasmid is a small extra chromosomal, that means it's outside the chromosome. It is usually covalently closed circular. In fact, for a long time, that was the definition of a plasmid, that it was covalently closed circular. More recently, I have read about plasmids that aren't. It's like, then how do we know it's a plasmid? The DNA can be recognized based on the nucleotide sequences at this point, what genes are on there. So how big, when I say these are small extra chromosomal loops? You see the hula hoop that's up on the cupboard? So you can see this loop, it's made up of different segments. If we think of that as being made up of genes, you've got yellow genes and red genes and blue genes. If you were to take that hula hoop and go stand beside the cross-country track, how many of you know there's a cross-country track on this campus? Well, there is. And the cross-country track goes from kind of behind where the swimming pools are, and it goes winding down the hillside, and then it comes and winds around, and then it goes down again and goes across a bridge onto the other side of the creek that's back there, winds over by the freeway and comes around, comes back. Anyway, it's very long. So if that cross-country track represents the chromosome, that would be the size of a plasmid by comparison. These things are really small, a very small extrachromosomal loop. And oftentimes there's more than one. These things reproduce independently of the chromosome, and because they're so tiny, they're using the same machinery that would be used to reproduce the chromosome, but they're going to reproduce much, much faster, and so sometimes there can be 100 or 150 of these things per cell. So they can have fairly high copy numbers. There are different types of plasmids that we'll be working with in this laboratory. Specifically, we're going to talk about F-factor plasmids. The F stands for fertility. F-factor plasmids are involved in transferring DNA from one cell to another. So they're involved in a process called conjugation. They can also be involved in a process called transduction. We're going to be doing, or transformation, excuse me. Transduction involves viruses. We do transformation later in the laboratory. We're actually taking plasmids out of one cell and putting them into another. So plasmids can be transferred either using sex pili, or they can be transferred without sex pili. And probably a more important type of plasmid is an R-factor plasmid. R stands for resistance. The R-factor plasmids are plasmids. They're separate loops of DNA, and they carry genes that make bacteria resistant to antimicrobial drugs. This has become extremely problematic because if one type of cell develops a resistance factor and it's on a plasmid, that resistance factor can be transferred to other bacteria, and pretty soon you have a whole bunch of different types of bacteria that are resistant. When antibiotics were first developed, so when penicillin first came on the scene, it was being used around the end of World War II. Penicillin became very commonly used. It was extremely effective against staphylococcus. And now it mostly isn't. Many, many strains of staphylococcus are resistant to penicillin. And the reason that this resistance, a lot of times it involves these plasmids that are carrying resistance genes. And they can be transferred from one type of bacteria to another. And they aren't. There's other types of plasmids. You don't need to know all the names of all the different plasmids. Just know what plasmids are. These small extrachromosomal loops of DNA. Well, okay, then there's ribosomes. What are ribosomes for? Protein synthesis. Whether we're in eukaryotic cells or prokaryotic cells, ribosomes have the same function. They are the site of protein synthesis. Again, ribosome refers to these small bodies. Soma means body. Ribose is the sugar associated with the RNA. Ribosomal RNA and protein is what ribosomes are made out of. Prokaryotic ribosomes are 70S. Eukaryotic ribosomes were 80S, and I said the Svedberg units that made up the 80S were 60 and 40. We found out Svedberg units don't add up. Same is true with prokaryotic cells, because here the ribosomes themselves are 70S, but the subunits are 50S and 30S. 50 and 30 doesn't add up to 7. It's like, really? Svedberg units don't add up. They're just a measure of density. So again, they're the site of protein synthesis. Here we see a diagrammatic representation. The 50S unit is bigger than the 30S. These things in the prokaryotic cell are not built in association with a nucleolus because there isn't one. The ribosomal RNA is encoded by DNA, which is in the nucleoid, and then the proteins had to have been produced by other ribosomes. These subunits only come together when proteins are being made. Otherwise, they're separate. Now, prokaryotic cells also can contain inclusions. So notice, again, we're skipping all those membranous organelles. They don't exist in the prokaryote. But inclusions in prokaryotic cells often serve the function of organelles. These things they're not considered organelles because they're not surrounded by unit membrane. That is lipid bilayer with integral and peripheral proteins. But they are sometimes surrounded by a layer of protein. So there are some important inclusions that you need to be familiar with. And they do have specific functions within cells. Here you see some, two on the outside are electron photomicrographs. The one in the middle is an image that you can see with the light microscope. So, first on our inclusion list are structures called carboxysomes. How many of you know what carboxy stands for? Are you familiar with carboxyhemoglobin? There's oxyhemoglobin and carboxyhemoglobin. What does hemoglobin carry? Oxygen and carbon dioxide. Carboxy refers to carbon dioxide. So carboxysomes are bodies involved in doing something with carbon dioxide. Specifically, these are little structures that contain accumulations of an enzyme that is involved in fixing CO2. In biology, when we say that cells are involved in fixing, what it means is they're taking something from the atmosphere and they're bringing it in and tying it to an organic compound. Ultimately, there may be some kind of inorganic thing in between, but in this case, they're bringing CO2 in and they're connecting it to a sugar molecule, a 5-carbon sugar to make a 6-carbon sugar. This fixing of carbon dioxide involves an enzyme called ribulose bisphosphate carboxylase oxygenase, or Rubisco. Here you see Rubisco. This is one place where I typically allow students to use this acronym. So Rubisco is the enzyme. Here you see the enzyme. The enzyme is accumulated inside these protein walls. And the protein is shown here, yellow protein and red protein. Most of it's yellow, the reds are only at the corner. The significance of this, apparently, ribulose bisphosphate carboxylase oxygenase, that enzyme is not very efficient. So in order for the cell to efficiently fix carbon dioxide, the enzyme has to be sequestered. It's inside these little structures. So cells that have carboxysomes can fix, they can pull CO2 out of the atmosphere and use it to make sugar. Autotrophs do this. We can't do it, but a lot of prokaryotic cells can. They have to have carboxysomes to do it. It's another type of inclusion called a polybeta hydroxybutyrate granule, or PHB granule. These are essentially nutrient reserves. So PHB is a polymer. It's collected when cells have a good nutrient supply available to them. They'll often store nutrient inside in these big granules. They almost look like vacuoles or something. So if you see them in an image, they look like these big open spaces. But these are accumulations of nutrient material. And they're actually visible with the light microscope if the cells are stained properly. So those are PHB granules. This is showing PHB granules, again, stained in slightly different ways. These apparently are used, people that are trying to get bacteria to produce a particular type of protein. Sometimes protein will be accumulated in those PHB granules, and then those can be extracted from cells. The other thing, I believe these things were partially responsible for bacteria depositing fossil fuel, which they did play a role in fossil fuel laying down of fossil fuels. This also mentions them being used in the production of biodegradable plastics. So people are trying to use inclusions of bacteria for different things. Another inclusion is called a metachromatic granule. These are phosphate reserves. So there are accumulations of phosphate in the form of polyphosphate. You have a bunch of phosphates stuck together. When you have additional phosphates stuck on a nucleotide, remember adenosine triphosphate? We've got 3 phosphates stuck on a nucleotide. That's a high energy compound. And that accumulation of phosphates is a big deal. So these polyphosphate reserves are considered energy reserves. The cell has a lot of phosphate, then they can make ATP much more readily. Metachromatic granules typically will show up as a color that's different from whatever stain you're using. So on the left-hand side, you see cells stained green, but the metachromatic granules are red. In the other two, the cells are supposed to be blue and the metachromatic granules red, but it's really hard to see. They're not very big, so they are difficult to see. And then some prokaryotic cells have gas vacuoles. I've also seen these called gas vesicles. They're just, again, a region where gas is accumulated. It's surrounded by a protein layer. It's not surrounded by cell membrane. So it's just a little layer of protein. And these things are involved in regulating buoyancy. So cells, if they have a lot of gas in them, they'll tend to float toward the surface in a water environment. And then if they don't have gas, they'll tend to sink. So they can regulate their position in a water environment using gas vacuoles. And then there are magnetosomes. So I mentioned magnetotaxis. Magnetotaxis, where cells can move relative to magnetic fields. Apparently that involves magnetosomes. These are usually made of iron. They can show up as these little dark staining things inside cells. So here you see a whole row of magnetosomes. Only certain cells have these things. Cells that are sensitive to magnetic fields. And then there are sulfur granules. These show up in cells if the cells can use hydrogen sulfide as an electron acceptor. We're going to get into metabolism later on. We'll talk about what can cells use hydrogen sulfide for. If they can pull the electrons and hydrogen protons off of hydrogen sulfide to use it as part of their electron transport, then they can deposit sulfur inside. This also shows up as kind of refractal bodies that are visible with a light microscope. We have some cells in that plexiglass box back there that can do this. They use hydrogen sulfide as an electron donor, and consequently they will have deposits of sulfur in them. Now, beyond those inclusions, we have a couple of specialized cell types we need to look at. Only certain types of cells make these things, so we'll consider them specialized cell types rather than inclusions. The two on the outside images that we see here are endospores, and then the ones in the middle are kinetiospores. So we'll talk about endospores first. Endospores, so remember when last time we were working with our soil sample and we put the tubes in the boiling water bath up here at the front? So you put water in a tube, and actually the water was already in there. You put dirt in it, and then you put it in this boiling water bath. We were trying to enrich for cells that form endospores. So endospores, they're specialized cells. They're dormant structures. They're not reproductive structures. They're dormant structures that are only produced by certain types of cells. Very often the names you'll see, the genus names are Bacillus and Clostridium, but the genus Bacillus has been split into a bunch of related genera now, Lycinobacillus, Painobacillus, Brevibacillus, there's a whole bunch of them. So quite a number of different bacteria produce endospores. If you do a gram stain tonight, you may get to see endospores, and they'll typically show up as white spots. So those purple cells with the white spots are endospores. What's interesting about these things, they are dormant. Basically, their function is to allow cells to survive during periods of unfavorable conditions. So they're highly resistant to damage. There's a number of reasons. One, they contain almost no water. If you don't have water, metabolism isn't happening, because remember, water is required for hydrolysis and condensation reactions. Without water, metabolism stops. So these things are metabolically inactive because they don't have any water. They have fairly high levels of DNA, but almost no RNA. Cells have to be making RNA in order to make protein, and these guys aren't. They're metabolically inactive. The DNA is there, but they're not using it to make RNA. They're not doing much. They have fairly high levels of calcium and a substance called dipicolinic acid. This apparently has something to do with their resistance and also their reactivation later on. Each endospore is surrounded by 2 layers of membrane and 2 layers of wall-like material. So they have thicker coverings than a typical cell, and that's also going to help them to be resistant, at least to physical factors. So what kinds of things are they resistant to? Heat, pressure, radiation, desiccation, and even exposure to toxic chemicals, because they have 2 layers of membrane, and because they're metabolically inactive. Toxins don't bother them much. So they're these dormant structures, and what's really interesting about them is how long they can survive. Because apparently they can survive at least thousands of years and potentially millions of years, and maybe longer than that. We really don't know. You have these highly resistant structures that can survive for a very, very long time. So researchers think 500 to 1,000 years in lake sediments, millions of years in amber. Remember Jurassic Park where they were pulling genes out of mosquitoes in amber? And that seemed kind of far-fetched, but apparently not when you're dealing with bacteria. There are actually bacteria trapped in amber that could be reactivated if they were spore formers. The spores were still viable after millions of years. So these things are formed inside a vegetative cell. The process is called sporulation. If you've had any of you have had a botany class, you'll see that a lot of the terminology here is similar to what plants do. So plants like fern plants, moss plants, they produce spores. Those spores are reproductive. Endospores are not. But the process of making them is still called sporulation, and the spores are contained in a structure called a sporangium. So you'll see a lot of terminology that's similar to what plants do. In order for a cell to make a spore, the first thing it has to do is copy its DNA. So it gets 2 copies of its genetic material, the DNA, and then the DNA is separated, the cell membrane actually folds inward, and you get 2 pieces of cell material. This is just reminding you the DNA is covalently closed circular. It mentions in that one diagram that starvation will trigger spore formation. Maybe. Sometimes that's true, sometimes not. So the cell membrane forms this septum, and then one side of the spore basically engulfs the other side. The process looks like endocytosis to me. Like this side comes across and swallows this side. The portion that was going to be the spore got really little because it lost all of its water. It's called a phorspore, but it kind of shrinks down and it gets surrounded by membrane. And then layers of wall-like material are deposited. There's a cortex outside the inner spore, or peptidoglycan-like material outside the inner spore membrane, forming the cortex, and then another layer of material outside the outer spore membrane that forms the spore coat. I'm sure you're looking at all these steps and going, seriously? So there's a whole series of events, and then what happens is the cell deteriorates, and what's left is an exospore. So we can look at a diagrammatic representation of this. Here we see an endospore inside a cell on the left, a diagrammatic representation in the middle, and this image on the right shows a bacterial cell here without any spore. Here's a cell with a spore in it, so this is an endospore, and these things are all exospores. The cells die, and these spores can sit around in the environment for millions of years. And at some point, if conditions become suitable for them, they can grow into a new cell. So they're just allowing the population of bacteria to survive, not to reproduce or increase in number. Each cell forms one endospore, and one endospore can form one cell. So here's the entire process. The DNA is replicated. So you get 2 copies of the DNA within the vegetative cell, and then the membrane folds inward. This is not entirely accurate because it looks like there's only 1 layer of membrane. There's actually 2. The membrane folds inward. It's a double layer of membrane. And then one side engulfs the other side. This is going to be the spore now. Then the layers of wall-like material are deposited. Cortex outside the inner spore membrane. Spore coat outside the outer spore membrane. Sometimes there's another layer called an exosporium that's formed. Basically, this is a mature endospore, and then the cell degrades and what's left is an exospore. So it's a multi-step process of forming a spore. I see you're looking at that clock. We're almost done. Okay, so what can happen to an exospore? These things can sit around for an indefinite period of time, and at some point, when conditions become suitable, probably has to do with water availability, they will undergo germination. What other activity does that term apply to? What germinates? Seeds. So again, a lot of the terminology comes from plants. People used to think bacteria were plants. So when a spore, an exospore, is laying in the environment, conditions become suitable, it will undergo germination and grow a new cell. So here we see a spore undergoing germination. It's taken on water, it's going to extend outward. This is the coverings that were on it. It just comes out and it becomes a new cell. I guess I have to stop. I'm looking at the clock, it's like, we're almost done. That's okay, we'll finish up prokaryotic cell and then get into taxonomy next time. We'll catch this where we leave off. I can't go over because you guys have a quiz tonight.
Yep.
You'll be all stressed out, wanting to go. Do some last minute review.
I'm gonna down some coffee and I'm gonna come back and I'm just gonna